applications, and this study tries to fill the gap. And it is important to highlight that there are types of features of antibodies that are linked to this kind of protection in cerebral malaria. And the researchers involved children in Malawi, where malaria is very, very common, and they were actually looking at groups of two uncomplicated malaria that was a mild disease, and those who had cerebral malaria, that is with severe brain involvement. And they compared this using serology systems. System serology in simple means looking at the immune response from any angles on just how much antibody is there, but also what those antibodies can actually do to fight the infection. And what was tested, the researchers measured both quantity and function of the antibodies, including how well they attach to the malaria parasites to help destroy the infected cells. And they were targeting this PEM5-1, which is actually a protein found on the surface of malaria-infected red blood cells, which allows the cells to stick to the wall of the blood vessels, especially in the brain, leading to blockage and inflammation. And it binds to some specific receptors on the endothelium, like the EPCR and ICAM1. And these are just normal proteins on the lining of blood vessels. The malaria parasites uses to attach to them, and when this happens in the brain, it can cause serious damage. And the study found out that children who did not develop cerebral malaria had stronger antibodies than that could call for help from other parts of the immune system, such as the complement proteins and also white blood cells, to clear the infections or the infected cells early. And this complement fixation is when the antibodies attract and activate special blood proteins or the complement system that actually can break open the infected cells and mark them for destruction, a process called opsonization. And also the neutrophil recruitment refers to neutrophils as one type of white blood cells, and they are good, and that the good antibodies can call these cells to the infection site and destroy these infected malaria cells. So what's the difference between